Good morning. Welcome. My name is Tom Rando. I'm the director of the Eli and Edith Broad Center for Regenerative Medicine and Stem Cell Research, and really wanted to welcome everyone today to this wonderful event as marking one of the events of our 20th anniversary year here at the Broad Center at UCLA. So it's a great pleasure to see such a great crowd. Today's event was actually initiated by one of our members, Melody Lee, who had experienced a Women in Science event at Rockefeller when she was there that she thought was fantastic. She suggested we think of something like this, and so that's why we're here to celebrate women in stem cell science. So it's a real great opportunity. And I was reminded by an email from the university that this is actually March's Women's History Month, and that March 8th, day after tomorrow, is International Women's Day. So, and I guess there are events coming up at the university next week, so we'll see more about those, but it's a real opportunity for us to celebrate the remarkable achievements of the women in the Stem Cell Center. Before we begin, I wanted to just pay tribute to the native people of this land. So the Eli and Edith Broad Center of Regenerative Medicine and Stem Cell Research at UCLA acknowledges our presence on the traditional, ancestral, and unceded territory of the Gabrielino Tongva peoples. As a land-grant institution, we pay our respects to the traditional land caretakers of Tovangar, past, present, and emerging. So we have a great program. I don't want to take too much time, but I wanted to just give a great thanks to the BSCRC staff who put this all together and everyone who would make this a really successful day. So let's give them a round of applause. So we have many VIPs in the audience, and a couple have agreed to say a few words before we get started. It's my pleasure to introduce Chancellor Frank, who is a leading global health researcher, and he's held positions in government and academia in both the US and in Mexico. And he became our 7th chancellor on January 1st of this year. Before joining UCLA, Chancellor Frank served as the president of the University of Miami. He was the dean of the Harvard T.H. Chan School of Public Health, and he was the Federal Secretary of Health in Mexico, where he implemented a groundbreaking program for expanded access to health care for 55 million uninsured citizens. So a really remarkable achievement. He's got a long list, of course, of achievements and accolades, and I won't go through all of them, but one pleasure of introducing a new friend and colleague is you find little tidbits that you don't know about them, and I'm going to tell you one that I didn't know, I bet most of you didn't know, and that is he's written 5 novels for children explaining the functions of the human body. So Chancellor Frank is a member of the American Academy of Arts and Sciences, the U.S. National Academy of Medicine, the National Academy of Medicine of Mexico, and El Colegio Nacional, the most prestigious learned society in Mexico. He's received honorary degrees from 11 institutions. Please join me in welcoming Chancellor Frank.
Good morning to everyone. Thank you, Dr. Rando. It's really a great honor for me to be here as I start my 3rd month on this job, and it's been quite a first few weeks on the job. But someone was telling me, thank you for coming here. We know how busy you are. If I wasn't doing things like this would not be a very attractive job. It's when actually I get to see what you all do that makes everything that happens here so worth the effort that we all put into making this such the great university that UCLA is. And especially when we focus on the first ever Women in Stem Cell Science Symposium, and we celebrate this achievement of 20 years of groundbreaking research at the Broad Stem Cell Research Center. Because that's 20 years of pushing boundaries, it's 20 years of discoveries that actually change lives, and it's 20 years of bringing together brilliant minds, including, of course, many of you here in the room who are working to transform medicine. Of course, none of this happens alone, so let me start by issuing a few additional thanks. First, to the Broad Stem Cell Research Center for making today possible and for leading the way in regenerative medicine. To Dr. Tomas Rando, who has led the center since 2021 and is helping usher in the next era of breakthroughs here. To Dr. Owen Witt, the founding director of the center, who led several achievements from 2005 to 2020. and to Edith Broad and the late Eli Broad, the center's namesakes, whose generosity has been foundational in expanding our center, in recruiting world-class researchers, and in training the next generation of scientists. And of course, thank you to all of you for being here, all who support the work and who carry out the work of this great center. We're living at a time when science is moving at an astonishing pace, and stem cell research is really reimagining what we are. We're in the midst of a revolution in the health sciences that really offers boundless potential to improve the lives, the quantity and quality of lives of millions around the world. What makes this center so vital is not just the leading research, it's what happens next because of the scholarship that's carried out here. It is pioneering clinical trials that take discoveries from the lab to patients. It is pushing science beyond theory into hospitals, into clinics, into real-world treatments. I am one of the people who thinks that our mission as researchers and educators doesn't end when we publish a paper. That's a journey that starts. We have the duty to society to translate our findings into solutions that actually help people every day, and to shorten that transition from discovery to development to actual treatments. And I think this is very much in the ethos of this center. As you all know, the center has driven some of the most important medical advancements of our time. Dr. Donald Kahn's groundbreaking work in stem cell gene therapy has cured children born with deadly immune disorders. And UCLA led the first ever FDA-approved clinical trial using embryonic stem cell-derived cells to restore function in patients with macular degeneration. But here is the challenge that brings us together. Even as science races forward, not everyone has equal opportunity to shape the future of science. Women make up only 1/3 of researchers worldwide. Only 3% of Nobel Prize winners in science have been women. And even today, many young girls still do not see themselves in careers in the sciences. And this is a loss to all of us. Now, I feel strongly about this topic because I have developed a 360-degree view of women. First of all, I have a twin sister. And when you share your mother's womb with a woman, you learn to respect women. But not only that, I have 4 other sisters in addition to my twin, and only 1 brother. So my brother and I had no doubt of who are the better version of the human species. So those are my siblings. I had a very talented mother who was a classical piano concertist. And I now have one of my sisters who does that, and another who's a cello player. But then going further up, my paternal grandmother, who lived to the age of 106, was publishing second editions of some of her books at age 103. So I still see myself having a very long career ahead of me. And then continuing with the family side, I'm married to a very talented professor who's now joined the medical school, Dr. Felicia Knoll, who is both my wife and a very esteemed colleague, and who would have been here had she not been flying right now today to some other place. We have 2 daughters, and I'm very glad that one of them, the oldest, is pursuing a PhD in science. I'm very proud of that. I have had 2 wonderful women who have been my bosses. Dr. Gro Brundtland, the first woman, former Prime Minister of Norway, who was my boss at the World Health Organization, and Dr. Drew Faust, the first woman president of Harvard University, who was my boss when I was a dean at that university. And every day I interact with wonderful, wonderful, amazing women. And I do believe that breaking the barriers that don't allow women to fulfill their enormous potential, which I'm sad to say as a man, is usually much higher than ours. I think this is one of our absolutely imperatives. Diversity is not just the right thing to do, promoting diversity, it's the smart thing to do, because diverse communities are much better at understanding problems, and at devising solutions. So what the Broad Center is doing is absolutely critical because this is a place of connection. We're inspiring young girls from K through 12, training the next generation of scientists and championing our female faculty and leaders. And this panel that I'm looking very much forward, and I'm going to stop very soon so that we can move to the real reason why we're here. This panel is a perfect example of all of that. So as we celebrate 2 decades of amazing progress, let us also set our sights on the future, a future where the next life-changing treatment is developed because this center had the vision to pursue that, a future where the next generation of scientists, more diverse, more inclusive, leads the charge, and a future where breakthroughs are not just for a few, but for all. I am excited to see how this center continues to push the boundaries of medicine for the next 20 years and beyond. Thank you very much.
Well, thank you for those inspirational words, Chancellor Frank. It's my pleasure now to introduce another VIP, the Dean of the School of Medicine, Steve Dubinet. So Dr. Dubinet was appointed as the Dean on June 5th, 2023, so he's been in the job for a while now. He's the Distinguished Professor in Departments of Pathology and Laboratory Medicine, Molecular and Medical Pharmacology, and Medicine, and he's served many key leadership roles since he's been here at UCLA. He was the head of the Division of Pulmonary, and he really rose that division up to the point where it's been at the, I think, top 5 programs in the country, U.S. News and World Report, for many years now. He also was the head of the Clinical and Translational Science Institute, CTSI, which is a really critical institute for moving forward programs from the bench to the bedside and doing clinical trials at UCLA and with other institutions. His work has been on immunity and inflammation in the development of lung cancer. And when he was a fellow at Mass General, where we actually overlapped, we found out for a short time, he was a lead in the first clinical trial using tumor-infiltrating lymphocytes to treat patients with cancer. And this was a revolutionary treatment, and it worked in many cases, but not very well in lung cancer. And Dr. Dubinet has spent his career and continues to do groundbreaking research in trying to understand this role of immune system regulation and cancer therapeutics. And he's been doing that. He continues a very vibrant research program while running the medical school. So please join me in welcoming Dr. Dubinet.
Well, thank you, Tom, and Chancellor Frank, for your support, your leadership, and inspiring words. We share many things about our family and women, and at some point we can discuss that. You know, it's really a privilege to be here for the inaugural Women in Stem Cell Science Symposium and to celebrate with you 20 years of groundbreaking stem cell research at UCLA. At the David Geffen School of Medicine, we're deeply committed to fostering an environment of respect, of inclusion, and innovation, one that values unique perspectives and contributions and expertise of every individual. So our scientific progress really thrives on this collaboration and creativity and bold new ideas. One of the ways in which we accomplish this, and in particularly in stem cell medicine, is to take these ideas and not only write the textbooks of tomorrow, but transform patient care today. Stem cell medicine is transformative. It has the power for repair, regeneration, and to actually heal. And it is in the progress now of redefining healthcare as we know it. We're shaping the future of medicine as we work with stem cells, predict disease, and treat risk. For the past 2 decades, the Broad stem cell has been at the lead in discovery, in developing innovative therapies to prevent, treat, and even cure, as you've heard, devastating diseases. Our scientists are not only conducting life-saving research, they're actually changing lives. For the past 2 decades, the UCLA Broad stem cell research has led in all areas of stem cell research. And in addition, we've focused on training the next generation of scientific leaders. And we've had remarkable scientific mentors, some of whom are here today, who have participated in multidisciplinary mentorship training programs, so that we've had undergraduates, graduate students, postdoctoral fellows, junior faculty benefit from that training in stem cell science. And so under their leadership of these leading mentors, they've had the opportunity, the trainees have, in terms of clinical translation and being involved with community outreach, importantly, ethics, and very importantly, scientific communication. These are essential skills in the preparation for academia, medicine, and also industry. Since the founding of the center, they have trained 385 young scientists. This includes more than 200 UCLA graduate students, postdocs, and clinical fellows, 149 students in a program that has student interns from Cal State Northridge, and more than 30 UCLA undergraduates. This is a program that fosters curiosity and cultivates expertise and expands the pipelines of leaders in stem cell science. So at the David Geffen School of Medicine, we are steadfast in our commitment to supporting and advancing the careers of women in biomedicine. And today we celebrate the brilliant women leading the way in stem cell science. These are scientists whose research and innovations are addressing some of the most urgent medical challenges of our time. This morning we'll hear about personal journeys, those that have involved becoming leading researchers in their fields, both the challenges and the opportunities they have encountered, and the discoveries on the horizon that could transform the future of medicine. It's truly an exciting time for stem cell science and for the exceptional women leading us in this field and moving forward. So I'm honored to be here today with you to celebrate with you, and now let's welcome back Tom Rando.
Okay, on to the main event. So it's my pleasure to introduce the moderator of this session, Dr. Judy Gasson. Dr. Gasson was the director for 20 years of the Johnson Comprehensive Cancer Center here at UCLA, and under her leadership, it really flourished and became an international presence. Also, for our own reasons today, we're grateful that she was the founding co-director with Owen Whitty of the Broad Stem Cell Research Center. And anybody who was there, I was not here then, but anyone who was around then, tells stories of how instrumental Judy was in getting the center up and running and really getting it going from the ground up. So we owe Dr. Gasson a huge debt of gratitude. So under her leadership, the JCCC, the Comprehensive Cancer Center, really grew to international prominence. They were involved in many studies, including studies that contributed to the FDA approval of groundbreaking therapies like Herceptin and Gleevec. And so really a storied center in terms of cancer research and therapies. And speaking of women in science, Dr. Gasson, of course, is a distinguished scientist in her own right. She was very involved in her career and continues to be in terms of cytokine research. She was, I didn't know this, she was instrumental in identifying GM-CSF, this cytokine that's used widely clinically in patients undergoing cancer chemotherapy and radiation therapy. So a really storied history in her own right. She currently serves as senior advisor for research and innovation at the David Geffen School of Medicine, and she works closely with the Technology Transfer Office and a lot of up-and-coming researchers who want to translate their work either into companies or into the clinic, and Dr. Gasson really facilitates that. Also, for our benefit, to our benefit, she's a member of the California Institute of Regenerative Medicine governing board, so she has insight into the workings of CIRM, which of course is central to the functioning of the Stem Cell Center and what we do. So please join me in welcoming Dr. Gasson and our panelists to the stage.
Thank you very much for that introduction, Tom, and thank you all for joining us today. Good morning. We have a terrific panel for you with 3 preeminent stem cell scientists that you're going to hear from shortly. So without further ado, I'd like to ask each of them to introduce themselves and to tell you why they chose to do research in stem cell science. Aparna?
Hi, everyone. Thank you for organizing this great day, and thank you for moderating. My name is Aparna Bhaduri. I'm an assistant professor in the Department of Biological Chemistry, and my lab studies human brain development and brain cancer. And I was really interested in studying stem cells of the human brain because I think that the human brain is the most brilliant, amazing feat of engineering anywhere. And I think that understanding the stem cells that make the brain for the first time is one of the most enigmatic ways of really understanding who we are. And I also think that it's crazy that these same stem cells come back many decades later, and when they do the same thing, they can be so devastating when they create brain cancer.
Yvonne?
Thank you, Judy. Hi, everyone. My name is Yvonne Chen. I'm a professor of microbiology, immunology, and molecular genetics here at UCLA. When I was about to go to college, my mom, as you might expect, asked if I wanted to be a doctor. And I said, no, I want to be making the things that the doctors use to treat patients rather to be the doctor. And so I was fortunate enough to have actually trained as a chemical engineer and then stumbled upon ways to engineer T cells, which are white blood cells, for them to be able to recognize tumor cells and kill them. And so my lab has been developing CAR T cell therapies for cancer and had the opportunity to take them to the clinic here at UCLA. And there are now patients who are alive and well because of it, which is, by far the proudest thing I have ever done. And that is really how I got into, stem cell research. It's that it provided a way for us to treat diseases that we could not treat otherwise, and has a lot of potential to give us new solutions to problems that we have been facing for many years.
Great. Amander?
Thank you, Judy, and I'm so happy to be here. I'm happy that the Stem Cell Center is celebrating women and women in science. I was recruited to UCLA in 2006, so it's been almost 20 years since I've been on this campus. It's hard to believe. I was one of the early recruits to the Broad Stem Cell Research Center. I was recruited with 5 other outstanding faculty, and we're all still here. 4 of those faculty were women. And so from the very beginning, the Stem Cell Center has been celebrating the careers of women, lifting up the careers of women, and providing opportunities for our research programs to flourish. In my research, we're interested in helping infertile couples have a healthy baby. And we do that by using stem cell technologies to improve outcomes with in vitro fertilization. So this has been the work that my lab has done over the last 20 years now, almost. At UCLA, I'm a basic academic scientist. I'm a professor in molecular, cell, and developmental biology in the Division of Life Sciences. So I see my dean, Tracy Johnson, here in the front. And I'm really looking forward to the conversation today.
Great. So the first topic that we're going to discuss is the many professional demands that are placed on faculty. You have to design experiments, you have to write grants, give talks, supervise grad students and postdocs, and of course be of service to our university community and beyond. So we'll start with Yvonne telling us how she manages to juggle all of these things and achieve maximal productivity in her research.
So that's a challenging question. I think we all do the best we can, right? I think my approach is I basically figure out what are the things I absolutely have to take care of, right? And make sure I get those done. And if that means working on weekends, working to midnight, it is what it is, right? There are some special demands, I would say, for women faculty. And it's also context dependent. So I mentioned I trained as a chemical engineer. I started at UCLA in chemical engineering. And I was one of very few female engineering faculty members. And what that means is I am constantly being asked to be on committees, to go to outreach events. Not, it's out of good intention. They want a woman in a room so they can hear your voice. They want the students to see a woman on stage so they can see a role model. So it's all coming from great places. But it does place a lot of extra demands on you when you also have to be doing everything else that any faculty members need to do. And so my strategy basically is I decide, do I think it's worth it, right? Do I think it's important that those students see me And if my answer is yes, then I'll do it. And if that means, you know, having another pizza dinner, then so be it, right? Because to me, it's worthwhile, right? And it's worthwhile to me because I think it's important that, you know, there are new generations coming up, and so you kind of just have to do that cost-benefit analysis.
Great.
Yeah, so I think the cost-benefit analysis is definitely a part of it. For me, I was given some very good advice, again by a female mentor at UCLA, that it's really important to know where the boundaries are as well. And so it's very easy because science is addictive. We love it. That's why we do it. It's very easy for science to creep into every corner of your day. You take it home at nighttime. You sleep with it at nighttime. You wake up with it in the morning. This great idea comes. But it's really important also to compartmentalize as well. So to really focus, to compartmentalize, to give your everything where you can to science. But also it's important to have a life outside of science as well. I'm a mother of 2 children, a 17-year-old and a 12-year-old. And it's really important that I give them space in my life as well, and to give them just as much energy and enthusiasm as I give to my science too. So it's figuring out how to compartmentalize your day so that you can give everything to all these different facets of our very complicated lives.
Aparna, you're an assistant professor. Can you tell us a little bit about the importance of mentorship in achieving this balance and achieving the goals of your research and your personal life as well?
Absolutely. I think that mentorship is really central to the scientific enterprise, and I've been very lucky as a graduate student and a postdoc to have mentors who really put me front and center in terms of the work that I was doing so that I was able to share my ideas with the world and really gain that independence. I've been trying to take that same philosophy to my own students, and I'm very lucky that I have an incredible lab which is primarily composed of women and other underrepresented individuals. And so it's really an opportunity for me to try and prioritize that mentorship and play it forward. I also think as a junior faculty, it's important to take information from different people, and both my chair and my faculty mentor, who I see in the audience here, are phenomenal in giving me some of that guidance, but also in giving me some of that space to make some of your own mistakes. And so I think that it's really a push and pull in terms of thinking about navigating some of these questions of really where do you put those boundaries and how do you prioritize the science and the trainees that you are here to mentor while also engaging in all of the other interactions that are really important for the functioning of the university and the department.
And I think the Stem Cell Center has been particularly good in making sure that people have the kinds of mentors that they need. And of course, as we advance in our careers, we then become the mentors to the next generation, which is really quite an exciting part of what we do.
If I may just add a quick point
Please.
about mentorship. I think this is a Women in Science event, but I think it's really important to emphasize that I think all of us have had great mentors that are both men and women, right? So it's not that you can only have great mentors who are the same gender as you. And you can also be a great mentor to someone who is not the same gender as you, right? I think it really is, you know, finding good mentors and being good mentors when you're in that position to do so. Very, very well said. One of the things that the Stem Cell Institute has been so effective in doing is providing the types of high-end research tools and infrastructure and so on that you don't want to have in your own laboratory, but you want to have available when you want to do your experiments. Can you talk a little bit about what is the role of philanthropy in stem cell research, and what types of things do philanthropy provide that you can't write on a grant or get through some other mechanism?
What I have really appreciated about philanthropic support for scientific research is that it enables you to form partnerships with other scientists that you may not have been in partnership before, and to think of something really transformative and perhaps not as predictable. And so you can sort of test ideas out, and then when they start to work and formulate, then you can apply through the more traditional funding mechanisms for grant support for that great idea. It's very difficult to get financial support from the traditional mechanisms where we get them from the National Institute of Health, from the National Science Foundation, unless that idea is pretty well formulated. There's some pretty good preliminary data that demonstrates it's going to work, and you've already done the first 3 experiments out of perhaps 10 that show that you're on a path to get there. So the place where philanthropy fits is to get those first 3 experiments done, is to do something really outrageous and crazy, and if it works, it can completely change a field. If it doesn't work, well then at least you tried it. You really put yourself out there and you tried it. So that's where I feel like it works well.
And building on what Amander was saying, I think that it really opens up the possibility to explore risky ideas. As I mentioned, my lab studies stem cells both in development and in cancer. And one of the ideas that we and others who think like us in the field think is that there's many different stem cells in the brain cancer. But this goes against the paradigm of how people have looked at brain cancer for the last 40 years. But those people who have had this other idea and unfortunately haven't made significant progress towards curing it are also the people who are reviewing grants for when we are applying through more traditional mechanisms. So having an opportunity from other sources, whether it's foundations or philanthropy, to explore some of the riskier ideas really allows us to get those first few experiments to show that this shift in paradigm really is the direction that the field needs to be going.
And I'll add that I think Amander and Aparna have spoken very elegantly about the beginning of the scientific process, right? The risk-taking, the preliminary data. But then you get through the bulk of your research, and then you get to what we call the valley of death, which is you have done the proof of concept, and now it's time to get it to the clinic. But getting to the clinic can take a couple $1 million, right? And there are no NIH grant big enough for you to do that. And investors don't want to invest just yet because you don't have any clinical data, right? So it becomes a chicken and egg thing. And that really is another place where philanthropy, in my personal experience, has played a huge role. So the only way we were able to get our first CAR T-cell therapy into the clinic was through philanthropic funding, right? First from the Parker Institute, and then from Steve and Jean Kaplan, who were generous donors to our cancer center. And that really allowed us to get the clinical trial going, to start dosing patients, and the data came out beautifully. And only then do commercial sources come in, because now it's like, oh, this works. Yeah, let's give you money. Right? But you need to somehow get through that valley of death in order for the therapy to have a chance. And so I think that's also really important to keep in mind.
That really echoes what Chancellor Frank was saying, is at a great public institution, we have a responsibility to go beyond the laboratory and to translate and, if possible, to commercialize our discoveries for the benefit of not just Los Angeles, not just the region, but for the entire world. And so that leads to our next question. Part of the reason we're enthusiastic about studying stem cells is that they provide the promise of a cure for diseases that we can only treat the symptoms now. So the 3 of you are all doing research that's very relevant to human health and disease. So I'll start with you, Aparna. Tell us a little bit more about how your research can impact health and disease in the brain.
Sure. We're very excited about this, and we really want to be able to take our findings to really treat brain cancer in particular. We think that in some of our work, we've been able to find new cell types that are in these brain cancers that we think are driving the cancer forward. And so by taking a new angle on understanding how these individual stem populations are working in the cancer, we think that we might be able to develop cures. The same understanding of these stem cells, I think, has a translation in the developmental context as well. These are some of the same populations that during development can go awry and can lead to vulnerability to neurodevelopmental disorders or neuropsychiatric disorders. And so while that might be a little bit behind in terms of translation, I think that the systems and the abilities that we have to look at human cells will have translational potential in the next 5 to 10 years, both in the cancer space as well as thinking about developmental disorders.
So the point that you're making is very important. We're not just thinking about stem cells as treatment, but also as creating the models that will allow us to find the treatments of the future.
Exactly. Yes, thank you.
Of course. Yvonne?
Yeah, so my group works on CAR T-cell therapy. So T-cells are a type of white blood cells that we all have, and what we can do is genetically modify them to express a new protein called a chimeric antigen receptor, or CAR, that allows them to recognize tumor cells. So my lab now, in the early days, my lab and many other labs have worked on CAR T-cell therapies for B-cell or blood malignancies. And now we're focusing our energy on solid tumors, particularly glioblastoma, which is the most common type of primary brain tumors in adults and is incurable. There is no actually effective treatment for glioblastoma. And so we're working very closely with our clinical partners here at UCLA. This is a great thing about being at UCLA. The hospital is literally across the street from my lab. And so we're working very hard on developing new therapies that we hope can be much more efficacious than what is currently available.
Can you walk us through how you make a CAR T cell, please?
Yeah, so there are different modalities. We do what's called autologous CAR T-cell therapy. That means the T-cells come from the cancer patient. And so I see Sugna and Dawn here, who are the leaders of our GMP facility here on campus, which is where we make these cells. So what happens is patient comes into the clinic, they go through a leukapheresis step, which is similar to if you went to donate platelets in the blood donor center. We take the white blood cells out of the patient, we transduce them with a virus that is not pathogenic, but it introduces the genetic material that's necessary to encode for the CAR, the chimeric antigen receptor. We then grow these cells out in the GMP facility, freeze them, make sure they pass all the release criteria, and then we reinfuse them back into the patient here at Ronald Reagan. And we monitor them very closely and to see how well the T cells are expanding, how well the tumors are shrinking. And so far, at least for B-cell leukemia and lymphoma, it's working pretty well. Yeah.
And how do the T cells know where to go in the body and what to attack?
Excellent question. So the CAR does not direct the T cell to anywhere specific. Rather, the T cell needs to naturally encounter the tumor in some way. In the case of B-cell malignancies, B cells are in the blood, so they happen to be in the same physical space as the T cells that we infuse into the patient. So they actually have an easier time. When the tumor is a solid tumor, a mass that's growing somewhere in the brain, in the pancreas, et cetera, then the tumor needs to be able to send out alarm signals, inflammatory signals that attract the T cells there. Now, in the case of glioblastoma, we can actually cheat a little bit. We can put the T cells straight into the brain. Other groups have done clinical study in human patients to show that that can be more efficacious. And that is indeed the strategy we're going to take with our upcoming clinical trial.
Which is very important. Glioblastoma is a deadly disease. And the fact that you're working on new strategies, these patients frequently have a survival of a year or less. So this is very, very exciting and important work. Amander, tell us a little bit about what you're doing and what you're trying to achieve in terms of wellness and patients and families that are trying to conceive children.
So what I really love about UCLA is it provides the opportunity to build something of impact. So I remember being an assistant professor, starting my lab. It's all about recruiting students and hoping students will come and work for you because there's so many other exciting scientists that they can choose from. Getting in research funding, getting in external funding, going through that process, going through tenure, and then becoming an associate professor and then ultimately full professor. So during my career over these 20 years, not only do I function and practice as an academic scientist and train graduate students, postdocs, and undergraduates in my lab, but I've also had really important administrative and leadership opportunities to start thinking about where does stem cell science fit, not just for training, but also in clinical care. And so I'm very excited about where stem cell science is going right now with regard to cures for devastating diseases. And in particular at UCLA, we already have some phenomenal science that is leading to cures of patients with devastating blood diseases. But something that I've been thinking a lot about, and I was able to start making inroads in this when I was in a leadership position at the International Society for Stem Cell Research, is thinking deeply about how is it that we're going to be able to afford these curative therapies. So right now, the stem cell therapy that's been approved to treat sickle cell has a price tag of $3.5 million. This is a very expensive therapy. And we're very lucky because this is a disease that does affect a number of people in the United States, but it's not something like heart disease that affects everybody. So what happens when we get a curative therapy for heart disease? And that has a price tag of $3.5 million. So how is our current healthcare system going to afford these new therapies? And so what we've been doing at the international level is start thinking about how do you put together a task force of people who are interested in ensuring that these amazing therapies that are coming out of labs at UCLA, other labs around the US, around the world, are going to be available to patients. How will they be available to somebody who doesn't have health insurance? How will they be available to someone who does have health insurance, but health insurance doesn't cover this single-shot curative therapy? Because our current healthcare system is actually based upon somebody taking a medicine for the rest of their life. I think this is where we are now in stem cell therapies. It's not just the amazing science that we're doing, that all of us are doing, but also thinking collectively about how can these therapies be available to everybody who needs them, not just those who can afford it. So that, I think, right now is some of the most impactful work that I'm doing, and I'm bringing my knowledge as a scientist to thinking about this problem, which I hope we can find a solution for in the future.
Do you want to say a little bit about your new institute?
Yeah, so again, at UCLA, this is what's so fabulous about being an academic scientist, is that I was given the opportunity by the Broad Stem Cell Research Center, by my dean, Tracy Johnson, by the School of Medicine, to build a new center for reproductive science, health, and education. I see Stephanie, who is our associate director, who is in the audience. So being able to build a center within a UCLA campus is a really wonderful privilege that I've been able to do. I built this center because I felt that there wasn't enough discussion or education around reproductive and women's health concerns at the undergraduate level. I also felt that there wasn't enough research being done around reproductive and women's health at the graduate student and postdoctoral level. There's very few labs who are thinking about these sorts of questions on the UCLA campus. So in building this center, what we've been able to do is transform the undergraduate curriculum and thinking about reproductive and women's health. We've been also holding public engagement exercises around the science of women's health and reproductive health. We've had hundreds of people attend our public symposia. And we're also starting to recruit, I'm hoping we'll be able to recruit this year, new faculty who are interested in the area of reproductive health and women's health and science. So I don't know if there's another place I would have been able to build a center like this, and UCLA has provided a wonderful opportunity to do it. I'm very grateful.
Well, I don't know about you guys, but I'm quite inspired hearing the stories of these, my fellow scientists here. And I know we have grad students and postdocs in the audience, and they're thinking about their career trajectories. So our last question for the panelists is, what would you say to a grad student or postdoc who's sitting in the audience today thinking about what they want to pursue in terms of their academic career?
Sure, I can start as the most junior person here, and I would say go for it. I think that stem cell science is one of the coolest frontiers, both in terms of understanding how biology comes to be and being able to look at it in a dish, and then being able to take it to the clinic the way that Yvonne is and others here are as well. And so I think that it's really a great opportunity. I also think that the world of science is changing rapidly. We have new AI tools that are coming in and are changing a lot of how we think science and work will typically be done. And I think that stem cell science is going to benefit from those tools, but also is going to need people who are thinking about the questions and really understanding the biology, because it's a new enough field that we don't know everything about it. And so until you have a substantial amount of knowledge, it's hard to predict what is actually going to happen with stem cells and how they work. And so I think it's one of the most exciting frontiers, and I think that there's really great resources here at UCLA and in California to really pursue those questions.
Yeah, I guess I'll comment on career in science more generally, right? I think it is just a fact that this is not going to be the easiest way to make a living. There are other jobs that will pay just as well or more, where you don't have to work quite as hard, okay? But I would say I would not choose a different career now that I've done it for a while. And the reason is it really is a privilege that we get to work on problems that we think are exciting, that we think are important, and we can come up with an approach that we think is the right way to do it and try it, right? That really is a privilege. And it's really exciting to be able to do it. Now, things don't always work out. I'm sure those who have been in the lab already know that, right? It's not an easy job. It's not an easy career, but it is so worthwhile. And so I would say, go do it.
I think the graduate student experience is one of the most transformative experience for a young person. I remember my PhD advisor saying to me, if everyone had a PhD, if getting a PhD was easy, everyone would have one. And getting a PhD is not easy. It is one of those times in your life where you have to struggle and you have to dig deep. And then when it comes to a completion, you've got this product that you can be proud of for the rest of your life. And so whether you stay in science and keep going on that track or move into the science adjacent fields, you've always got that experience. And so I chose to stay in science because I wanted to have an impact. I couldn't have imagined the career I would have had. Judy and I were talking earlier, I grew up on a sheep farm in outback Australia, and we didn't even have a movie theatre in the town that I grew up in. To now be here on this stage, being able to talk to you what it's like to be a scientist at a #1 public institution in the United States. So you should definitely go for it. You never know where your path's going to lead.
There is truly something magical about having an idea, designing an experiment, and in that moment to be the very first person that has a bit of knowledge about how the human body works. And I completely agree. It's a great privilege to be on this stage, to be at UCLA, which I believe has one of the most collegial and collaborative and supportive environments where you can work together in the Broad Stem Cell Institute and achieve more than what you had perhaps even imagined would be possible. And I know that it's our goal as the more senior people here to try to create an environment where everybody can exceed beyond their wildest dreams. And it's really a pleasure to be here today. We want to take a few questions from the audience, and we have just about 10 minutes left, and so we're more than happy to answer any questions that any of you may have for the panel. Oh, good. I see a hand. Yes, please.
Good afternoon.
I think we have a microphone coming. Thank you, Nusha.
Good afternoon. My question is immune resistance a critical factor in terms of the efficacy of stem cell treatment?
Can you repeat it again?
Yeah, immune resistance. Is that a factor at all in terms of the efficacy of stem cell treatment?
Do you want to take that?
I can start commenting specifically on CAR T-cell therapy, which is only one type of cell-based therapy. And the answer is yes and no. Okay, so in the context of autologous CAR T-cell therapy, because the cells came from the patient, the cells themselves should not be rejected. But we do genetically modify the cells with a protein that is not natural. And so the patient can actually develop some resistance to the CAR itself. And some patients do. Luckily, what we have found is that, at least in the context of B-cell malignancies, we tend to be, let me be very specific, in the context of B-cell lymphoma, if we can manage to wipe out the tumor and keep the patient tumor-free for about a year, they tend to do very well. The survival curve tends to flatline, as in patients don't die. And so even if they develop resistance down the road, it seems to be okay because we actually managed to get rid of the tumor. Now, the same thing may not apply for other indications where the tumor has a tendency to stay dormant in the patient and then emerge later on. So immune resistance definitely is something that we keep in mind, and there are strategies to try to avoid that. For example, we try to quote unquote humanize all the proteins that are non-natural that we put into a cell to try to make them as non-immunogenic as possible, right? To essentially try to hide them from immune resistance mechanisms.
Really quick question. When you mentioned white blood cells before. Is that equivalent to natural killer cells as well?
A very good question. No, natural killer cells are a different type of blood cells. So T cells are the ones, they basically function very differently. So T cells have naturally occurring T cell receptors that are specific to certain proteins. Natural killer cells kill in a slightly different way. Their dominant killing mechanism is to detect anything that is non-self. So in a sense, you can think of it as T cells are looking for something that shouldn't be there, okay? Like a bacterial protein, a viral protein. NK cells are looking for things that should be there, and if they're missing, then they know something's wrong, and NK cells would try to kill them. I'm like grossly oversimplifying things, but that's kind of how I would describe it.
We have another question from the audience.
Hello. I'd like to start by really thanking you all for speaking today. I'm a undergraduate student, so I'm still new to the field, but this was very inspiring as I kind of figure out what I want to do. My question is specifically to Dr. Chen. And so I mentioned that I am kind of new to the field, but your work with CAR T-cell therapy is really inspiring, and also with the solid tumors. I kind of wanted to ask what your lab, or how your lab is approaching the solid tumors.
Yeah, thank you for that question. So, how much time do I have? Let me be very, let me be very clear. So there's some major problems or challenges of solid tumors, right? Number one, how do you target solid tumors without killing healthy tissue that we do not want to kill, okay? So that's two very important components. One, being able to recognize the tumor, and number two, recognize the tumor while sparing healthy tissue. And the third issue is how do you get to the tumor, which is a question that came up earlier, right? Like how do you get the T cell to find the tumor? And once they find the tumor, now the tumor is a solid mass, how do you penetrate into the tumor? And so what we've been working on is what we call armoring the CAR T cells with various soluble factors and membrane-bound proteins that would overcome these challenges. So just as an example, we've developed CAR T cells that secrete a novel protein that can help normalize the blood vessels, the vasculature around the tumor cell, to help the T cells get to the tumor more easily. We've armored the T cells to secrete very potent cytokines that allow them to not only the CAR T cells to attack the tumor, but also recruit naturally occurring endogenous immune cells to come wipe out the tumor cells that the CAR T cells themselves cannot recognize. And so we're taking sort of a multi-pronged approach to try to improve the efficacy. We also, at the same time, need to be mindful of toxicity, right? We can make super, super potent T cells that also kill the mouse. And we don't want to do that, right? And so it's a balancing act.
There's a question, yes, right there. Thank you for your patience.
Hi, this is for Dr. Chen also. You have to get rid of the cancer first before you can restore functioning. So when you have cancer that metastasizes, you have to get rid of the cancer first, like the stem cell might be able to repair the functioning. So how do you envision working in the future with AI and clinical trial to try to speed up and get you an answer sooner about whether you're on the right track or need to go in a different direction? Because as you know, cancer doesn't wait for anybody, and we're losing too many good people.
Yeah, thank you very much for that question. I think AI is already being used at different levels, okay? So at one level, it's, for example, antigen discovery, figuring out what target on the tumor cell to go after. Okay, and that helps in terms of being able to digest very large data sets to identify what is unique on tumor cells and not present on healthy tissue we don't want to target. I'm also personally sort of experimenting with AI at a protein engineering level. Just last year I did a sabbatical and happened to be in David Baker's lab, who won the Nobel Prize in Chemistry
Good call.
last year. Yeah. I can't take any credit for that, but yeah, anyway. And so here we're using machine learning and AI to design new proteins that can do things that would otherwise be impossible to do because there are just no naturally occurring proteins that do it. Now, let me just be very practical here. AI cannot solve all the problems, right? So one easy problem is actually what was mentioned earlier. When the protein is designed computationally, completely synthetic, it can very well be immunogenic, right? And so oftentimes there are other problems that we have to solve when we use tools such as AI and machine learning, but it definitely is now being actively integrated into all of these therapy development pipelines to try to help us solve these problems faster.
I think we have time for one more question. Yes, right over there. We could otherwise stay all afternoon, of course. But.
Thank you so much for speaking to us today. This is a question for all of you. Given the current climate around science in our nation today, do you have any concerns now, specifically as stem cell biologists, that your field specifically will hold in the coming years? And then do you see any challenges for women and minorities to come specifically in these fields as well?
I'll take the first shot at that, and I'll say we're very lucky to be in California because the California Institute for Regenerative Medicine was approved for $5.5 billion for stem cell and gene therapy research, and there are about $3 billion left. And so people who are doing work in this area have been and will continue to be very well funded. It's fully state funded, there's no federal dollars there. Another source of funding comes from the intellectual property that we generate, that we use to translate and commercialize the ideas that are born in the universities that Chancellor Frank talked to us about. And so certainly devastating cuts to NIH would have an impact, but we're also a very creative and resilient group of people. So we're very hopeful that the worst case won't come true. And we need to continue to emphasize, like my colleagues did today, how stem cell research is impacting patients and families, and how important it is to keep pushing ahead for these cures for people who have the devastating illnesses out there. Other thoughts?
I would echo everything that Judy very eloquently said, and I would say that this is really something that will be a challenge, but I think that it's also an opportunity for us to have and take initiatives to speak to the public and tell them why our work matters, why is it that what we are doing is so important, and how can we get them involved and excited about what we're doing. So I don't want to minimize the challenges and the uncertainty, but given that nothing specific has yet really come to pass, I think that we need to be creative and optimistic and also really still keep encouraging the next generation, because they really are also going to be the future, and we can't give up on them now. We can't pull back on our support for them. And so I think that we really need to dig in and just be creative in how we get through it.
I think your question is a really good one because it's recognized and acknowledged that trust in science has diminished over the last 5 years. So I think this is a really important conversation. And there's lots of reasons for it, but in part, as scientists, we haven't done a really good job of explaining what a scientist does or what the scientific process is. And this was most illuminated during the recent pandemic we've all gone through, where as scientists, we knew you get a piece of information and you make a decision. You get a new piece of information, you might have to actually change the decision you've just made in light of this new piece of information. We understand that's the way the scientific process works, but the general public didn't. And as a consequence of that, they don't know if they can trust us when we say something anymore. And I think that's really unfortunate. So we have opportunities now. Now that we've learned this, we have opportunities as scientists to be able to go out there and be better communicators with the general public about what is it to be a scientist, what is science, how does science evolve and change with new pieces of information. So there's real opportunities there at our level, and also with the way we teach our students too, and have our students in the lab and part of our scientific mission.
So I'd like to thank you all for coming today, for participating in this event, for sharing the excitement and the enthusiasm around stem cells, and for celebrating the 20th anniversary of the Broad Stem Cell Research Center. So thank you very much for coming.